Hello guys, welcome to my YouTube channel. So in this series of lectures on cell biology, today we are going to see the details of transport of molecules from the nucleus into the cytosol. So first see the structure of the nuclear envelope. Now nuclear envelope contains several pores on its membrane which are called as the nuclear pore complexes or NPCs. So as if you can see in the right hand side of the figure, here this is the structure of the nuclear envelope, and on the nuclear envelope you can see several pores has been shown. So these are called as the nuclear pores or the NPCs. Now these nuclear pores or nuclear pore complexes, these are made up of several proteins called as the NPC proteins or the nucleoporins. So these NPCs, nuclear pore complexes, are made up of several proteins called as the NPC proteins or the nucleoporins. There are roughly 3000 to 4000 NPCs within a typical mammalian cell, and there are very high rate of traffic within NPCs. So the NPCs transport 500 macromolecules per second in both the directions at the same time. Shorter molecules pass easily through the NPCs than the larger ones. The larger the molecule, the more slowly it traverses through the NPC. Now, if you can remember, within each transport molecule within an organelle, suppose if we are interested in looking at the transport of a nuclear protein, so what will be the characteristic of a nuclear protein? This is the nuclear protein, and at the N terminal it will have a signal sequence which will guide this protein to the specific destination. In this case it is nucleus. Now this signal sequence, here as you can see, there are several types of signal sequences based on where the proteins need to be translocated. Here we are discussing only on nuclear transport, so we will only concentrate on that. For example, if there is a protein which needs to be imported within the nucleus, then its signal sequence must have this sequence of amino acids. As you can see, it must be rich in lysine and arginine. Likewise, you have several other signal sequences. For example, this is the signal sequence of mitochondria, plastid, peroxisome, endoplasmic reticulum. So depending on which organelle the protein needs to be transported in, there are several types of signal sequences. Now, there are certain nuclear import receptors. These import receptors are soluble cytosolic proteins that bind both to the nuclear localization signal on the protein to be transported and to the NPC proteins. So these import receptors bind both to the signal sequences present on the protein to be transported within the nucleus as well as to the NPC proteins. Here as you can see, this is the nuclear import receptor. This is the cargo protein which needs to be translocated within the nucleus, and this is the nuclear localization signal. Second figure has been shown. This is another cargo protein which needs to be translocated. This is the nuclear localization. This is the nuclear import receptor, and this is the nuclear localization signal. So the nuclear localization receptor basically binds both to the nuclear localization signal as well as to the NPC proteins. These NPC now one thing, if you remember, the NPC proteins which make up the nuclear pore complexes, these NPC proteins contain large quantities of phenylalanine and glycine, and so these are also called as FG repeats. Why? Because if you have studied one letter amino acid code, F stands for phenylalanine and G stands for glycine. So the NPC proteins which make up the nuclear pore complexes are rich in phenylalanine and glycine, and these amino acids are called as FG repeats. FG repeats serve as binding sites for the import receptors. So these are the places where the import receptors bind on the NPC proteins. The receptor cargo complexes move along the transport path by repeatedly binding, dissociating, and then rebinding to adjacent FG repeat sequences. So this is the this is the cargo receptor plus cargo protein, and this binds to one FG repeat present on the NPC proteins. So now how this complex of the cargo protein and the receptor is translocated. First it binds to one FG repeat on the NPC proteins, then it dissociates, and then it rebinds to another FG repeat. So in this way, this complex as a whole with the cargo protein and the import receptor moves from one place to another. In this way, the complexes hop from one NPC protein to another to traverse the tangled interior of the NPC proteins. So now we will see one GTPase which plays an important role in the transport of cargo proteins from the nucleus and cytosol and vice versa. So Ran is a monomeric GTPase protein that is found both within the nucleus and the cytosol, and it is essentially required for the transport of molecules within and from the nucleus. So as you can see on the right hand side, this is the Ran GDB. Okay, so this is the Ran monomeric GTPase, and here when in the cytosol it is bound to GDB. So when this Ran moves from the cytosol into the nucleus, it gets converted into Ran GTP, and afterwards when this Ran GTP moves again back to the cytosol from the nucleus, it gets converted into Ran GDB. Like other GTPases, Ran is a molecular switch because it can exist in two conformational states, one bound to GTP and the other bound to GDB. So remember, GDB is bound only when it is in the cytosol, and GTP is bound only when it is in the nucleus. There are two Ran specific regulatory proteins. Number one, cytosolic GTPase activating protein gap that triggers GTP hydrolysis and converts Ran GTP to Ran GDB, and the nuclear guanine nucleotide exchange factor GEF promotes the exchange of GDB for GTP and thus converts Ran GDB to Ran GTP. So as you can see, where Ran GDB is present within the cytosol, when it enters the nucleus from the cytosol, what happens? This Ran GF, guanine nucleotide exchange factor, converts the GDB to GTP. As a result, it is converted into Ran GTP. Now again, when this Ran GTP enters the cytosol from the nucleus, Ran GAP, GTPase activating protein, converts the GTP back to GDB. So in this way, the cycle of conversion happens, and because Ran can exist on two conformational states, it is called as a molecular switch. Now this Ran, this GTPase, does not only alter between the two states. What happens actually, in this way, it transports the cargo protein from the cytosol to the nucleus and vice versa. So the proteins which needs to be transported from the cytosol to the nucleus bind to this Ran GDB, for example. So suppose there is a protein P which needs to be translocated from the cytosol to the nucleus. So it binds with this Ran GDB, and as it enters within the nucleus, it enters along with it, and in this way that protein is translocated to the nucleus. Okay, this is the nuclear import mechanism. If we have another protein which needs to be exported, for example, so what will happen? It is here within the nucleus, and this protein P1 needs to be exported out of the nucleus. It will bind to Ran GTP, and in this way it will be ejected out. So suppose you need to export a protein from the nucleus to the cytosol. So suppose this is the nucleus, and here this side is the cytosol. So as you can see here, this yellow one is the cargo protein and this blue one is the nuclear import receptor, and here you have the Ran GTPase bound to GTP. Okay, so what will happen? This Ran GTP will take along with it this complex of the cargo protein and the nuclear import receptor, and both will be simultaneously translocated from the nucleus into the cytosol. So once they have been translocated, this cargo protein is released from that, and Ran GTP is converted into Ran GDP now because it is within the cytosol, and this receptor is also released. So this process can take in the reverse direction also. Suppose you have to transport some cargo protein and from the cytosol to the nucleus. So what will happen? The complex of the cargo protein and the receptor will bind with the Ran GTP, and it will get translocated from the cytosol to the nucleus, and then the cargo protein will be released within the nucleus. Ran GDP will be converted to Ran GTP because it is within the nucleus, and the import receptor will also be released. So here I have shown a comparative analysis of how nuclear import takes place versus how nuclear export takes place. So here, as you can see, suppose you need to import a protein, what will happen? This is the protein. This is the protein that needs to be imported, and here within its tail you can see this is a signal sequence SS. So it first binds to the nuclear import receptor, this blue one. So first binding with the nuclear import receptor with the protein to be translocated, this takes place, and then this complex of the nuclear import receptor and the cargo protein binds to Ran GDP because you need to transport a protein from the cytosol to the nucleus. Within the cytosol you have only Ran GDP. So the cargo protein along with nuclear import receptor binds with Ran GDP, and in this way this translocation takes place from the cytosol to the nucleus. So once within the nucleus, the cargo protein is released. This is the cargo protein which has been delivered to the nucleus. The nuclear import receptor is released, and Ran GDP is converted to Ran GTP. Okay, so this is the process of import, nuclear import. Suppose you need to export some protein, then likewise you have nuclear export receptor for them. Okay, so what will happen? This is the cargo protein which needs to be delivered to the cytosol, and here this is the tail portion, this is the signal sequence. So first this cargo protein will bind to this nuclear export receptor, and this complex will bind to Ran GTP. Okay, so here they have bound to each other because within the nucleus you have Ran GTP, and then it will move along and get translocated to the cytosol. Afterwards the cargo protein will be released into the cytosol, the nuclear export receptor will be released, and Ran GTP will be converted into Ran GDP because here we are in the nucleus now. So this is the comparison between nuclear import versus nuclear export.